So before going into more details, so I would like to preempt this talk by saying that some of the Hulk biology or Hulkology that I'll be talking about was initially defined by Dr. Sebastian Alvarado at Stanford University, and I've expanded on that to basically talk about cancer specifically. So some Hulkology for you. So the Hulk's been around for quite a long time. It first appeared in 1962, and the premise of the Hulk is that Dr. Bruce Banner, when he gets angry and stressed, he changes and transforms into this massive giant beast of a creature called the Hulk. Now the Hulk is very strong, very fast, his body can heal very rapidly, and apparently he's ageless, and he can do all sorts of other cool stuff. The Hulk is green, yeah, and also more unbelievable than anything else is got 5 to 7 PhDs. I mean, why would you put yourself through that? I don't know. Right, so before we go into Hulk biology, we first have to talk about cancer. Now, the reason we have to do this is because cancer is such a horrible disease that affects a lot of us. And the very stark stats are that 1 in 2 of us will develop cancer in our lifetimes, for those of us born after the late 60s, 70s. Now, that's very stark news, but the good news is that through advances in understanding what cancer is, how we diagnose it, how we treat it, more than half of these people will live longer than 5 years, and this number is increasing. So good news there. So why is this change? So why do we now have people that have cancer is no longer a death sentence? Why do people live longer, live better lives, and we can treat and diagnose cancer better? And this is because we understand cancer biology better. So one thing I would like to note is that first myth to break. Cancer is not one disease, it's actually a collection of diseases that has similar characteristics. And in the early 2000s, two scientists, Doug Hanahan and Bob Weinberg, came up with the idea of conceptualizing what makes a cancer. And they came up with the idea of the hallmarks of cancer. Originally, they said it was 6 characteristics, and then a bit later in 2011, they expanded that into 10, which is what you can see here. And what you can see here is these 10 characteristics, which are either biological characteristics or context that makes cancer a cancer. Now, I don't have time to go through all of them today. I do go through in the book chapter, I do go through most of these, or nearly all of them, but today I'm going to be focused on a few of them. And the first one we're going to be talking about is, for me, the start of it all, which is mutation. So what I want you to do now is to have a quick think. What's the first superhero, or your favorite superhero, or the first one that comes up to your head? Okay, so you probably came up with one of the 10,000s of superheroes that are available to choose from. Now, my favorites are, because I'm a child of the 80s, Ninja Turtles and X-Men, particularly the 80s cartoon series, which is way superior to any films that we have nowadays. So the common characteristic between Donatello, Gambit, and Rogue is that they're mutants, and they either were born with mutations or they acquired mutations that made them their superheroes. And this is a similar origin story to a lot of superheroes. So to understand that, we need to think about, okay, so what is a mutation? Because we use that word, we hear that word a lot, but what does it actually mean to be a mutant? So a mutation happens when in a gene, you have a permanent change to that genome. Normally there's damage to DNA, that's permanently incorporated in the genome, and the mother cell can pass that when it divides into daughter cells, and so on and so forth. Now, the reason why this is important is because genes code for proteins. And if a protein is mutated, it can either be changed in their structure, so it can have a different shape, it can be shorter, it can be not made at all, or it can be either active all the time or not active all the time. So its function and its structure basically changes. And this is really important because cancer is a disease, a genetic disease, and it's underpinned by mutations in the genome, and particularly acquisition of a series of mutations throughout progression to proper full-blown cancers. So what causes mutation? So I already mentioned DNA damage. So our cells are exposed to thousands of DNA lesions every day. By existing and living, we are exposed to tons of DNA damage. So when our cells use oxygen to break up sugars to make energy for our cells, there's production of what we call reactive oxygen species. So that means that there'll be active oxygen species that can damage DNA. When we walk in the street, we get exposed to UV light, that can damage DNA in our skin cells, and so on and so forth. Those of you that smoke, and you shouldn't, that can cause damage to the cells in the lining of your mouth and, and to your lungs. So there's a lot of ways in which either inherently to our biology or to the lifestyles that we choose that can damage our DNA. Now, if cells weren't able to cope with that DNA damage, I don't know why that keeps happening, I'm sorry. If we couldn't cope with DNA damage, we wouldn't have evolved beyond single cells or organisms. And so our cells are uniquely able to detect and fix DNA or deal with it, DNA damage or deal with it. So what happens when a cell is exposed to DNA damage? So for example, X-rays, what can happen is that the cells can detect that lesion, they can recognize it, and then some specific molecules will tell the cell, now repair the DNA, and if it's repaired, you can keep on dividing, or if not, then cells self-destruct in a process that we call apoptosis. And again, is one of the hallmarks is that cells can't do that anymore. So what are the molecules that actually do this? So one of them, and it probably is the most important protein you never heard of, is called P53. Now, P53 is what we call a tumor suppressor gene, and is commonly known as the guardian of the genome because it's so important to regulate this response to damage and whether cells can fix it or eventually die. And actually, we know that if cancers lose P53, either completely or because it's a mutant that doesn't function, that the majority of cancers lose P53 function and they will then progress. So what happens in the case of the Hulk then? So let's go back to him. So I already mentioned that Bruce Banner can change from the Hulk to the Hulk, and that is caused by, originally in the origin story, by Bruce Banner being exposed to a nuclear blast, which exposed him to gamma radiations, which is a type of ionizing radiation that damaged DNA. So that should have killed him, but it didn't. So what happened after that is that he realized that when he gets angry, he becomes the Hulk, and then when he's no longer angry, he comes back to Bruce Banner. So gamma radiation, ionizing radiation, damages DNA, and is actually one of the ways in which we can treat cancer, because we use X-rays and ionizing radiation as radiotherapy to kill cancer cells. Because in such high doses, cells can't cope with the amount of damage, so they'll never be able to repair it, so they always commit to death. So Bruce Banner should have died, but he didn't. So what happened? What happened was that a process called chromothripsis, which is when the chromosomes in a particular cell get chopped up into tiny, tiny bits when exposed to catastrophic amounts of damage, in this case, nuclear blasts and gamma radiation, and then get kind of quickly cobbled back together. And this happens in cancer when sometimes there's massive changes in breakage of chromosomes. So what happened is that this rearranging of chromosomes in Bruce Banner's cells led to him be able to change from the Hulk to Bruce Banner to the Hulk. So for example, gaining the ability to grow very fast, to heal very fast, and to deal with the stress associated with that change. So how does that happen? So I already mentioned that Bruce Banner becomes the Hulk when he gets stressed or angry. And what we can speculate is that the stress hormones that drive that change, what they could be doing is they could be binding to receptors or proteins inside the cells and then driving the expression of these changed genes that instead of normally just making him a bit annoyed and, bang a table forcibly, it actually makes him change into a giant green monster. And that, in a way, that can happen in tumors, because what you can have is that they get changes in the way the genes that normally would be controlled in terms of promoting cell growth and expansion into in growth and proliferation are normally off, and some genes that are normally turned off are on. So in cancer, you get this switch. So things that normally should be off are turned on, so cells just keep on growing without control. And things like P53, which I mentioned before, which can regulate whether cells will continue dividing if there's damage, are turned off. So cells will keep on dividing even in the presence of damage, and that will mean that they'll be more likely to acquire mutations and then actually become a full-blown tumor. So once the Bruce Banner becomes the Hulk, so how does the Hulk survive or manage to exist with all that bulk? Because now we have an enormous mass of muscles, green muscles, moving around. So it requires a lot of energy. And this happens as well in cancer. So when cancer grows very, very fast, and when it grows so fast, it uses up all the sugars it can get, so it uses up all the glucose it can get, but also it grows away, from blood vessels, which means that the supply of oxygen and nutrients will drop very quickly. Particularly, so this example here is a bowl of cancer cells called a spheroid, and I think they're breast cancer cells, if my memory is not mistaken. And what happens is that oxygen can only diffuse up to 150 microns through a tissue. So beyond that, the levels of oxygen start dropping. And we know that these drop in oxygen levels lead to changes in biology, which can allow the cells to adapt to low oxygen conditions. And so one of the things that can happen is that cells will change from oxygen-dependent energy production to oxygen-independent energy production, which is analogous to what happens when you go for a run and your muscles start getting a bit burny. So that what happens when the oxygen levels, so you can't breathe fast enough, particularly if you've been fit like me, you can't breathe fast enough to supply oxygen to your legs, and then the cells switch from this oxygen-dependent to oxygen-independent biology, energy production, and then one consequence of this is the production of lactic acid, which causes that kind of burny sensation, which is probably one of the reasons the Hulk's always so angry as well, because he's basically just aching all the time. So we've now talked about a bit more about the Hulk biology. So low oxygen biology can change the Hulk in many ways as well. So as it changes cancer, it can allow recruitment of blood vessels. So I told you they grow away from blood vessels. So low oxygen biology can lead to attraction of these blood vessels back to regain better oxygen and nutrient supply. It can also help a lot of the things that I can go on about because I'm a hypoxia geek. Right, so, but next thing, so this bowl of cells here is artificially green. So my student dyed this green with a fluorescent dye, but the Hulk is actually green. So what does the greenness mean? What's the importance? What's the origin of the greenness? And how is this relevant to cancer biology or biology of the Hulk? So the theory, again, that Sebastian Alvarado postulated is that, and one thing I would like to note is that in the Hulk mythos, it can be green, but it can also be purple-ish gray as well. So this is important. So what could happen is that when he grows so fast, you got rupture of small blood vessels in the tissue, so small capillaries in the muscles when he grows so fast. That means that red blood cells get released. Then you have processing of hemoglobin in the heme groups into these two molecules down here, biliverdin and bilirubin. Now, what biliverdin and bilirubin do is that they're the molecules that give the purplish green color to bruises. So when you bruise, you got rupture of little blood vessels, red blood cells get destroyed, and you get production, accumulation of biliverdin and bilirubin. So that's potentially a reason why Hulk is green or purplish gray, depending on what part of the story you're looking at. But how is this important in terms of our cancer biology model? Well, in our cancer biology model, biliverdin and bilirubin can actually protect the Hulk against the consequences of this change. Because when it changes, you got this increased metabolism, it's using up all this energy, you got inflammation going on, you got all these potentially very stressful conditions, cellular stressful conditions that can damage DNA. And you don't want further damage, even the Hulk doesn't want further damage and mutation. So what biliverdin and bilirubin can do, it can actually have anti-cancer properties and reduce some of these cancer-promoting conditions like inflammation and reoxygen action species in damaged DNA. So they actually in the lab been shown to have cancer protective properties. So again, being a bruise, massive bruise, is actually good for the Hulk. So finally, I already told you about several aspects of cancer biology using the Hulk as a model. I didn't touch about all of them, and in the book chapter, I managed to talk about 9 of the hallmarks in the context of the Hulk, and managed to justify how they'll map onto the Hulk. But there was one of them that eluded me, and that's the 10th hallmark. And that for me is kind of the end point. So if mutation is the start, the 10th one is kind of the end point. And this is the reason why most people die of cancer, which is the invasion and mutation, invasion and migration metastasis hallmark. So this is when cells can escape the primary tumor, spread throughout through the blood circulation and go colonize a distant organ. And the majority, 90% of patients die of this type of secondary cancer rather than the primary tumor itself. And this is why we really need to understand that biology of metastatic disease. And what we have here is, again, our green ball of cells, again, green not biliverdin, is just fluorescent green. But what we did is put this little ball of cells into a layer of gelatin-like molecules that kind of mimic what surrounds tumor cells, the tissues in the tumor. And then what we can see is that very rapidly they start moving into these spaces. And this is analogous of what happens in the body. So this aspect of the hallmarks of cancer is the real true supervillain of the story, not superhero. So with that in mind, and after all this whole ecology and whole biology, the only thing we couldn't figure out is, how do these trousers stay up? Nobody can understand that. So right, so I think that's me. So if you have any questions, just let me know. And yeah, I hope you enjoyed the talk.
Thank you very much for this nice talk. For me, I am not at all in that area, so it was a little bit difficult to follow everything. And I just have one quick question before everyone else can
go for it.
You said that the tumor will eat a lot of sugar and everything to grow. If we stop giving them sugar, what will happen then?
So actually, there's therapies. What they do, you don't stop giving it sugar because you can't shut down nutrients in your body, because obviously it's in the context of an organ. But what you can do is there's drugs that can switch off the tumor being able to grab the sugar. So the cells in their surface have little transporters that can transport in sugar into the cell to be used up. So one of the therapies that focus on this kind of energy metabolism are switching off these adaptations to kind of take up as much sugar as possible and starving out the cancer cells. So you don't take it off, you don't reduce the amount, so diet wouldn't impact it on it because homeostasis would not allow that to happen. You need drugs to switch off that at the cancer level.
Yeah, because when we talk about sugar, it's not real sugar, it's sugar.
Yeah, it's glucose and other very, very simple sugars, yeah. But other types of nutrients can be used as well. So cancers can adapt to start using up fat. They can use amino acids. They can use a lot of different things once the metabolism starts switching. So as with anything in cancer biology and science in general, is a little bit more complicated than that.
Yeah, and is it you know, there is priorities in the body that which organ will hit first, as for example the brain.
Yeah.
Is the tumor also prior number one, or how does it works?
In terms of, well, what the tumors can do. So one of the things that low oxygen biology can do is it can upregulate, so switch up a lot of these things that makes the cell more likely to get the sugars. So it tries to mop it up. And this is one of the reasons why in cancer patients, particularly when they're very sick, you get this thing called cachexia, which is when the body's basically eating itself. So people get very thin because they're basically using up as much as it can because it's just growing so fast and so out of control. So it's not a matter of priority, but it will take up as much as it can to grow.
Yeah, thank you.
Yeah.
I will let other people to ask a question. Yeah. Maybe Gonzalo, you will have one, or maybe Florian.
Well, maybe in the meantime, I can ask, since you work on hypoxia, how does that relate to tumors and cancer?
So hypoxia. So hypoxia biology is an adaptation to low oxygen conditions that can happen in normal physiology. So like I said, when you exercise, or when you go up a mountain, or it can happen in other diseases like during stroke or heart attacks, anywhere with oxygen is deprived from tumors. From oxygen. But in a tumor, because they grow so fast, you get these regions of low oxygen. And basically the tumor utilizes these biological changes to allow it to grow better and to spread. So, you know, all the 10 hallmarks I mentioned, pretty much every single one of them, low oxygen biology can drive. So it can increase genomic instability and mutation because it can lead to things like P53 being removed from the genome. It can do the energy metabolism switch. It can help to recruit blood vessels. It can do all sorts of things. And it can also increase the expression of molecules that will make cells more likely to spread and colonize this organ. So any of the hallmarks, hypoxia, biology. So there's not low oxygen itself, it's the biology driven by low oxygen conditions, which is what won the Nobel Prize for Medicine and Physiology last year. So in the hypoxia field, we're all like, woo, excited, we're validated now. It was very good. But yeah, that's how it drives it.
I also have one question for you, Isabel, for the very nice talk. I really enjoyed the superhero analogies. Since I used to work in research of regeneration.
Oh, okay, cool.
They were also quite popular.
Yeah.
during the talk. It's more like a generic question, but like as you pointed out, the cancer it's not one single disease, and we often hear about certain types of cancer that are much harder to treat than others. What are the main features that make these cancers harder to treat? Is it a matter of being hard to diagnose, or does it also have to do with certain properties of the cancer specifically?
It's a combination of both. So in terms of harder to treat, so some cancers are just harder to diagnose. So things like one good example is pancreatic cancer, ovarian cancer. Because of the way they grow, and I think it's because of the position of the abdomen, people don't have symptoms until it's very late and it's already starting to spread and grow beyond the point in which we can control it. That's where they really have, unfortunately, really high mortality rates. Whereas a cancer that grows like in the skin, you can see a mole growing. Or, for example, bowel cancer, there's very clear symptoms of that. Lung cancer, again, coughs and things. So late diagnosis is a big issue with making some cancers more like having higher mortality. But being harder to treat is another one. So sometimes when I talk about cancer, people go, oh, when do you have the cure for cancer? It's like the cure for cancer is such a vague thing because each cancer is its own unique thing. Even from patient to patient, even within the same tumor, different areas of the tumor will be more resistant to therapy than others. So again, another mistake we think is that cancer, because I show these images of a ball of cells and they're all the same cell type, but actually the tumor, first, you don't just have cancer cells, you have immune cells, you got support cells, you got all sorts of other things that can promote cancer, but also even the cancer cells, they can have different genetic makeup. So they have different mutations because they evolve. Again, and there is cancer evolution, so they will evolve slightly differently in different populations within the tumor. So where there's one, for example, could be really sensitive to chemotherapy, but then the rest of it could be very resistant, and then it overtakes and it grows even stronger. So the challenge we have with cancer treatment is the cancer biology itself, but in a case-by-case basis. So yeah, and for example, other things that might make a cancer harder to treat is the location. So brain cancers are really hard because you have to, for like x-rays in the brain, it's hard, surgery in the brain is hard because the brain is such a sensitive organ that you can't just, go at it. And like you can remove a chunk of the colon and you'll be all right. You can't really remove a chunk of the brain and be all right. Or, you can remove a chunk of the liver and you'll be all right. So location can be an issue as well. So where they are in the body. Yeah, so there's a lot of reasons. But again, a cancer is multiple diseases, not just one disease.
Yeah, we exactly have that question on Facebook, if we will ever find a cure for cancer.
Right. Okay, this is a difficult one. So I could say that there's already are cures for cancer. Surgery is very effective depending on what stage the cancer is found. Chemotherapy and radiotherapy can be very effective depending on what stage and what tumor type it is. The thing about cancer is that there's not going to be a single one treatment thing. It's not like a vaccine in which you have and that's it. You've now cured the disease. Cancer adapts. So even what you need, even if one cell remains, so if you do like chemo, radiation, surgery, and there's one cancer cell left, that one cancer cell is enough to make another tumor. It might stay there for a long time and take a while to go back, but it will grow back eventually. The other big issue is what I was mentioning at the end, is this issue of tumors spreading to other parts of the body. If you catch a tumor early on, they're very treatable. So, you can resect a very localized colorectal tumor, and the patients are probably going to live very well for the rest of their lives. Like my grandfather had a tumor, he didn't even have to do chemo, it was fine. But with some cases, the big problem is when cancer spreads. And the problem with cancer spreading is that sometimes it spreads and people don't know it is spread. So, for example, breast cancer is a good example. Breast cancer, the primary tumor, if caught at an early stage, if it's not very aggressive, it can have really good prognosis, and women can take various types of continuous therapy that will keep any tumor growth at bay. However, the problem with breast cancer is that sometimes it has spread already and people don't know it, and it can take 10, 20 years for the secondary tumors to start growing. And by that stage, it's a bit too late. So one of the big challenges in the field more obviously treating cancer is really important, but actually diagnosis is fundamental, because the earlier you catch it, the more likely your therapies we have or more you might develop will be to be effective against it. And also another thing in the field is to what we call personalized medicine. And it's not just the cancer field, it's in other fields as well, which is, again, taking each cancer as an individual entity and then personalizing the patient and also following it up. So things like imaging of cancers can allow us to follow up and see how the cancer is responding through therapy. So you can change it as you go along. So adaptive radiotherapy, for example, is very important because you can change as the treatment goes to see how the tumor is responding. So again, there won't be a single cure for cancer. We already have surgery and chemotherapy, radiotherapy are super effective. They're the most effective we have. But immunotherapy is coming along really well. And also small molecule treatments, so very targeted, that target very specific nitty-gritty biology. This can be great in terms of reducing side effects, but they're very context dependent. So they depend on the tumor that you're looking at. So we need to have really good screening tools to be able to know which patients will respond from those treatments. And it might just be that in 100 patients, 2 will respond, but those 2 will respond amazingly and they will never have a tumor again, but it might not be suitable to the 10. So designing clinical trials that will test that properly is a huge challenge.
Yeah, so what is the minimum size of a cell to be detected? Because if it's spread, then could it be very, very small that we cannot see it, or which tool do we have for screening for
Right, it depends on the tumor type. So it depends. So for some tumors, you don't even have to have cells. So things like detection of PSA in the blood for prostate cancer. So you take a blood sample and you check for this thing being produced. For some others, like mammograms, it needs to be, again, I'm not a radiologist, so I'm a cell biologist, but it needs to have enough they can be detected by imaging. And the big limitation of imaging is that resolution, is how small can you go. So this is where things like PET imaging, which uses traces which are not just images of the tumor like a CT scan where you just take you look at the contour of the tumor, but you're actually using a labeled tracer which can look at, basically boost up the signal for a particular biology. So we would look at, cancer cells eat up a lot of glucose. So you have this radio-labeled glucose that's taken up by the cancer cells. So if you scan the patient, even really small deposits can glow up like a light bulb, and then you will know. So it can pick up, for example, secondary cancers as well. It does have some issues that are non-specific, like the brain will take it up. So because like you said, the brain uses up a lot of glucose. So the brain lights up like a light bulb. The bladder lights up like a light bulb because it gets secreted through that. So we detect it out. But it is one of the ways in which we can increase our ability to diagnose cancers earlier is through imaging. So better specific tracers, particularly things like PET and other these sort of radio tracers, will be really kind of the way of the future for imaging and detection of cancer.
Okay, because I thought PET is not very fine in spatial resolution.
Yeah.
MRI was maybe better, but maybe
not. So it's not in terms of resolution in there, it's more in terms of specificity of the biology. So that's why PET is cool. I mean, MRI, yes, is much more in terms of combining disciplines again. That's where we will go forward. But the cool thing about PET is that allows you to look at the biology and distinguish the biology of normal cells from that tumor cell. So things like, if you have a good PET tracer for metastatic disease, that'll be fantastic, because you can just then use it to look at, say, scanning a lung, and you can then see the deposits, and then you can be guided, see how far it's gone or whatever. So that's where it's very useful. But yes, you're right. It's not great in terms of resolution, but it's great in terms of the biology side. And this is where cancer biology and oncology goes as a discipline, is that we want to use the biology as a way to expand our knowledge, but also as a way to treat and diagnose it. Not just relying on the anatomy, but relying on the biology.
Thank you very much.
Yeah. This is my first Pint of Science pint glass.
Yes, great.
And I've got one from 2018, but this, no, 2019. I don't have one from this year because there weren't any, but
yeah.
that was just a talk just about the hallmarks of cancer. So that was a different one.
There's a question on the chat from Anna.
Yeah.
Taking into account the high cost of personalizing individualized treatments, would this be something available in the near future?
What, in terms of personalized medicine?
Medicine to treat cancer.
Yeah, personalized medicine is challenging in terms of cost. Now, there are good news. So, for example, whole genome sequencing is getting cheaper by the day. It used to be like we had the whole genome project in 2000. It was originally published, and that was costed millions. And now you can do whole genome sequencing for less than $1,000 or whatever it is. So the technology is getting there, but obviously still $1,000. In terms of drugs, a lot of it is to do with to get a good targeted therapies, it takes a long time. So it takes about 15, 20 years to go from concept to clinic, to actually being available to the clinic. And a lot of this process involves pharma companies. And again, they have to make their money somewhere. So that's why they hike up the prices. So in a way, one of the things that's quite exciting in terms of cancer biology and cancer therapy is to what we call improve the therapies they already have. So like improving things like radiotherapy, which is much cheaper. Improve the use of conventional chemotherapies and drug repurposing. So drugs which are off patent and they've been clinically tested, which can then be used more easily, and they're cheaper. So there's a combination of that. Yes, true personalized medicine is very expensive, but I think for me is the way that we need to go. And if it's personalized medicine in the way that we segregate the patient using the biology, but then treat them with more generic treatments, then there might be the way forward. So as an example, so in hypoxia, so hypoxia is not, like I mentioned, low oxygen conditions are known to be tumor drivers. So we know they're markers of poor prognosis. We know that patients that have hypoxic contents that respond poorly to therapy, they don't respond to radiotherapy very well. We know this, we've known this since the 90s. We've known this very well. But any drugs that have been tested to try and either hijack the hypoxic biology or reduce hypoxia, they've failed in the clinic a lot. We really haven't had success with hypoxia targeting or hypoxia related drugs. And the reason for this is that clinical trials are really badly designed because we don't have good ways of detecting what's the hypoxic content in patients to do that easily. The best thing we have at the moment is a probe they have to stick into the tumor to physically measure the oxygen tension. And that's not feasible for the majority of tumors. And it's very invasive and painful. So patients might not want to be exposed to that. So it's really bad design of clinical trials that held up drug development. And I think that's also the reason why sometimes they cost so much, because it can take a long time to get from concept to drug. But there are really cool ones they're coming up. So yeah, but they're expensive. I mean, immunotherapy is stupidly expensive, even worse than small molecules. So yeah, but these things eventually will become more readily available. I'm a big fan of targeted therapies. I think exploiting the biology, because I'm a biologist, so I'm like, if you can exploit the biology, that's so cool. It's like so cool. Like chemotherapy is like ugly, just kill everything. It's like so much more elegant to target things.
She says obrigada.
Nada.
Okay, I think we can stop now. We are at the end of the time. So maybe you have some words to say, Gonzalo, before we close today's edition for Pint of Science.
Yeah, I can just thank both of the speakers for taking our invitation to be here today. It was very nice to have you joining the event, and the topics are super relevant and interesting to hear about. And also to thank the people from the Stockholm team that together with me have been putting together events since last year, also in Stockholm, when we could still gather at a bar, and now temporarily online. So thanks for the help of everyone involved, and I hope the participants found it interesting, and stay tuned for tomorrow, because we have 2 more talks and 2 more speakers. So see you tomorrow.
Exactly. Yeah, exactly. I really want also from the Central team to thank you, the team in Stockholm who is doing an amazing, great job. So thank you very much.
Thank you.
And yes, tomorrow we will have 2 other speakers, and it will be Miguel and Anis. And so we are always at the same place, at the same time. So we hope to see you tomorrow. So I will stop recording now, stop sharing online. But of course, if you have any question, please reach us from the channel you want, email, Facebook, Twitter, directly on our email address. Yeah, please share your question. We are really open for discussion. So thank you so much for today and see you tomorrow.
Thank you, everybody.
Thank you.
Bye.
Bye-bye.